All right. Let's hope. Okay. Any questions about the things we were talking about on Monday, where we started talking about cancer and some of the kinds of damage that can lead to cancer, and the idea of oncogenes and proto-oncogenes and tumor suppressor genes being in balance? Any questions about that kind of thing? Okay. Let's pick up again then. We were getting to talk about base excision repair, and okay, I'll get that. Thank you. Unmuted. Thank you. And we're around. Should be around here. Okay. We were starting to talk about base excision repair as a generic process, and this is the generic description of what base excision repairs does. So there's a base that's been damaged in some particular way, and there's a base that's damaged in some particular way, a specific DNA glycosylase, and we're going to talk about a couple of the glycosylases, cuts off the base, leaving the phosphodiester backbone intact, but that leaves you with, whoops, hi. I started recording, I think. Okay, good. Hi. Yeah, I get nervous about that. I know how sad people are if it doesn't work. That leaves us with the abasic site, which is not good. And once it's lost its base, it just looks all the same, whatever it had been. So at this point, the endonuclease recognizes that as an abasic site, cutting it open, so there's an opening space. And then polymerase and ligase are going to have to come in, add a new base, close up the gap. And we started talking a little bit about a couple of specific glycosylases. We talked about the 8-oxoguanine glycosylase, so it can recognize the 8-oxoguanines specifically to cleave those out. And I think we were just starting to talk about uracil DNA glycosylase. Uracil, we mentioned, is one of the more common mistaken bases and so forth. It happens if a cytosine accidentally is oxidatively deaminated. The structure is in the phosphates of the phosphodiester backbone have negative charges, and they can help to stabilize the polar deoxyribose as the base is taken off. This empty site, this abasic site, is going to have to be dealt with, and there's a specific kind of an endonuclease that attacks there. There are a number of different kinds of endonucleases, and this one recognizes it as an abasic site. It attacks on the 5' side of the abasic sugar and then takes off the sugar. So it creates an actual gap in the DNA backbone that's one shorter, or just simply missing that sugar at that spot. And importantly, it leaves a free 3' hydroxyl group. And that means that because there's a free 3' hydroxyl group and a template across from it, DNA polymerase can act there. And so it can add on an appropriate base. DNA polymerase doesn't close the gap. That's done by ligase. Base excision repair is a base. It's just that base is going to be handled. And there are several bases that fall under the umbrella of base excision repair. Suppose you've got some other kinds of damage. Maybe you have more than just one damaged nucleotide. Maybe you've got a form of damage that there's no specific glycosylase for. An alternative way of fixing it then, apart from base excision repair, is nucleotide excision repair. And let's take a little bit of a look at how that would work. In this case, you have damage of some kind. Here they're showing it as a single nucleotide. Could be a little region even. The helicase opens up the helix. It opens it up into a kind of a bubble. And an endonuclease then can eat off a region around the damage. So it's not just the single base of the base excision repair. It's a region of phosphodiester backbone. This leaves an extended gap now. but the gap has a free 3' hydroxyl, so the DNA polymerase can fill in. It'll leave a little gap there, and the DNA ligase then can repair that gap. So now, nucleotide excision repair, involving this whole stretch of nucleotides being taken out, the enzymes for that were actually discovered through genetic diseases, which turned out, when people understood them better, to be, in fact, errors in nucleotide excision repair. There's a syndrome called Cockayne syndrome that has a number of different conditions associated together. That's what makes a syndrome. The neural development is not perfect. The individuals are shorter for height, for their age. They're sensitive to sunlight, so they react much more strongly to it than you or I might. And they age prematurely, so that they look, at 20, they might look 40 or 50. And it turns out that this whole syndrome, these common kinds of defects, can result from mutations in any of 5 genes. So there's sort of a constellation of 5 genes, any one of which, if they're mutated, can give rise to the same Cockayne syndrome. And what these genes share is that the proteins that they encode are involved in recognizing and removing stalled DNA polymerase at places where the DNA has been damaged. So if you imagine making RNA, and we're going to talk more about that in the next chapter, and the polymerase is rolling along and it hits an area of damaged DNA, it can stall at that spot. And it can't go forward, it can't go backward, it can't come off, it's just stalled. And the genes of nucleotide excision repair are, all of them, it turns out, involved in being able to recognize those stalled polymerases and releasing them at those sites. You have to get that polymerase off before the nucleotide excision repair machinery can repair the damage. One of the consequences of these gene defects is that there are more regions of unrepaired DNA than in the average person, and so there are higher levels of apoptosis. There are more cells that have DNA damage in them that gets recognized by the apoptotic machinery, so more cells are undergoing cell death than in a typical average unaffected individual. And this is probably behind the accelerated aging that's seen in individuals with Cockayne syndrome. Another set of enzymes for nucleotide excision repair were found through a different genetic disease called xeroderma pigmentosa. And you can figure out that with the derma, it has something to do with skin, and pigmentosum, that probably extra pigmentation of some kind. People with xeroderma pigmentosa are extremely sensitive to sunlight. Individuals with xeroderma may be 1,000 times more likely to get skin cancer than an average individual otherwise would. There are 7 different genes under the umbrella of xeroderma pigmentosa, and these genes are involved in repairing UV damage. Now, since the tissue of our body that gets the most UV is our skin, people with xeroderma pigmentosa are specifically vulnerable to skin cancers and not just cancers anywhere in the body. There's a variant form of xeroderma pigmentosa that's actually caused by mutations in a DNA polymerase. So there's a variety of DNA polymerases. You have the main one that's going to do the bulk of replication, but there's a little zoo of several other DNA polymerases that serve their own purposes. And one of the cute properties of polymerase eta is it can replicate across from thymine dimers, and it can replicate correctly across from adeoxyguanine. So polymerase eta actually can correctly replicate that kind of DNA damage. There are structural reasons for that. The active site on polymerase eta is a little bit more open and a little bit more flexible. You remember we talked about the thumb, fingers, palm, generic structure of polymerases. And so because it's a little more open and flexible, it can accommodate these damaged things and slip in the nucleotide to polymerize opposite them. In individuals who have this variant, they have defects in polymerase eta, so they are not able to replicate the DNA damage, and so they end up with a skin cancer as a consequence of that. So, but this brings up the general idea of translesion DNA polymerases. So if you have an area of damage, hopefully, many times, many times, base excision repair or nucleotide excision repair will repair that damage. But what if they don't? What if you've got damage that is not, in fact, repaired? These translesion DNA polymerases are a little cluster of polymerases that are capable of replicating despite the damage. But they are, for instance, here is REVRO-NP can polymerize across an abasic site. They're obviously picking at random what it should have been, but at least there's something there, and you can go on with the rest of the machinery. These are distinct from the bulk DNA polymerases, the main DNA polymerases, because they're more error-prone. They are not as faithful in their copying process. So you might ask, well, why would you want a DNA polymerase that makes errors? And the answer is that this is sort of a triage solution. When you have high stress levels and there's a lot of damage to the DNA going on, you may not be able to keep up with it with base excision repair or nucleotide excision repair. And under those stress conditions, these translesion DNA polymerases at least ensure that you don't get stuck with everything stalled on the damaged DNA, that you can continue the replication. You may have picked up some more errors in the process, but you can kind of carry on and hope to patch it up later on. What about gaps? So far we've talked about the kinds of damage where one strand of it has some damage and the other strand is okay. But suppose you have a break. Suppose you have, just a clean separation so the thing is broken apart. This is obviously not a good thing. You can imagine that places where there are gaps in the DNA are going to stop DNA synthesis. And so they're not a good substrate for replication. And so in some cases, not all cases, these can be recovered by processes of recombination. And so in recombination, you're going to use the information you have on other strands to help you repair the damage on this strand. In order to do recombination, you need a homologous DNA elsewhere in the cell to use as the corrected copy. We've talked about the fact that prokaryotes have circular genomes. I may have mentioned, but it may have been far enough ago you forgot, that many of the prokaryotes have more than one copy of that genome at any one time. So they may have 2 copies or 3 or 4 copies. So they may have a circular piece of DNA, but they may have more than one copy of it. Of course, in most eukaryotes, many life stages are diploid. They have 2 copies of everything all the time. And that allows for this homologous recombination. The machinery for doing homologous recombination is highly conserved among prokaryotes and eukaryotes. And so this says both that it's ancient and that it's essential. This is one of the important ways that cells can maintain the intactness, the integrity, the correct coding of their DNA. And what does that look like? Okay. Well, here, let's start with a single strand break to begin with. Here we have we're replicating at the moment. This is a replication fork going, and there's a gap. This is one that wasn't caught by nucleotide excision repair, for example. So here's a gap, and we're going to have to deal with that. So there's a helicase that can unwind the DNA at the gap. The free 3' end here can displace this strand, and it can then make the complementary region available to provide the missing information, like this. So if you kind of keep track of everything here, none of it is sort of flopping in the breeze at any one time. The helicase will unwind through there. You've got this free end here, and you've got the replicated stuff to there. That free end can dive down, hybridize, releasing that. And so now you can copy through there And we've resolved the strands, but we still have a gap, and we're going to have to deal with that gap. But we've been able to get past the gap. In other words, we can continue replication. We're going to have to deal with the gap still. And that gap can, again, be dealt with by DNA polymerase and ligase. But suppose you have two strand breaks. If you have both strands broken, this is a substantially harder problem to deal with. And what happens is the cell deliberately gets rid of some of the DNA it's got by using something called an exonuclease. So it uses an exonuclease to nibble back. It's going to nibble back several nucleotides to create a free 3' end for the left-hand strand and a free 3' end for the right-hand strand. Now, this is only going to work if there's homologous DNA around to borrow the information from. All right? So this second strand here is another double helix that is homologous to the one that was damaged. That is, it has, in general, the same sequence as the first one. On our chromosomes, our homologous chromosomes are often identical, but they're not always identical. But it needs to be pretty identical, close to identical. Now we have these free 3' ends, and we have some homologous DNA. And the free 3' ends can, I love the phrase, invade the homologous DNA. So this free 3' end is going to hydrogen bond to that half of the homologous DNA, and this free 3' end is going to hydrogen bond to that half of the homologous DNA. So now both 3' ends have something to be extended from that is the appropriate sequence for that DNA. So DNA polymerase now will continue to elongate. So this little arrowhead will keep getting longer and longer, and this little arrowhead will keep getting longer and longer. And DNA polymerase will continue until we get to the point where we have filled in all the missing information. This is still a little bit tangled, and there's a process called resolution where you have to swivel the different carbon backbones and so forth. And after you've resolved that structure, which may involve other proteins, you end up with two complete DNA sequences. Only in the top strand, you've borrowed the information from the bottom one there, and so forth. So what you have now is a molecule that is mostly like it was to begin with, but it had to borrow some information from the other homologous strand. So recombination is quite important. It's an important aspect of dealing with DNA damage, and it is an important aspect of sexuality. When you have meiosis, you have recombination at a certain stage of meiosis just to kind of keep the variety rich in the gametes. There are some proteins that are used in recombination that are present all the time. They're not just there during our S phase. They're there all the time. And one of these is Rec-A, or recombination protein A. This is a structural schematic of how Rec-A, or recombination protein A, coats DNA. You can see here the red double helix inside it and the kind of blobby sea slug-like things sitting on top of it. So what Rec-A can do is it can bind to double-stranded DNA. It will extend it and untwist it. It's expended by spending ATP, so it's an energy-dependent process. And that opens it up so those single strands can come in and invade. So what Rec-A does is it allows those free 3' ends that were generated from the damaged piece to get access to the intact piece. And we can tell that this is an important process in general because mutations in Rec-A lead to a loss of recombination and a condition called ataxia telangiectasia. Hi, is there a comment you'd like to make? Oh, okay, great. Another thing that can happen to double-strand breaks, so if you have a double-strand break, both pieces are there. We just talked about homologous recombination as one strategy for doing it. Another one is end joining. You've got two free ends. You just line them up and smush them back together again. The risk here is that you don't know for sure who you're doing it to, right? So end joining between two blunt ends can end up being mutagenic if you're in fact generating sequences that didn't exist before. Deinococcus is really good at this. That's one of the reasons you remember that picture of the little confetti of DNA at the end of irradiation for Deinococcus. Deinococcus is extremely good at blunt end rejoining, and there's some thought that it keeps its DNA kind of packed in lifesaver rolls of coaxial cable, so when there's a blunt end break, that ends are still close by. Okay. So we've talked so far about some of the kinds of DNA damage. We've talked about some of the repair mechanisms that allow us to do that. We know that in some specific genes that are involved in repair can in fact cause diseases. So let's look at some of the genes that are related to, more specifically to cancer, not just damage of DNA in general. And two of the places where we pick up a lot of these have to do with cell growth, not too surprisingly. There's something called the cell cycle. And in general, the life of the cell is a matter of growing for a while, going through DNA synthesis, growing for a while, going through mitosis, and then doing this over again. That process obviously has to be pretty well controlled for it to be done right each time, and there are cell cycle control proteins called cyclins. These are proteins that were first picked up because their abundance was very different at different times in the cell cycle. The other thing are stages in the cell cycle where there's kind of a quality control aspect where they can detect things, and there's specific signaling pathways for that. And here is a picture for a generic picture, if you like, for a cell cycle. You notice there's a G1 phase, an S for synthesis, a G2 phase, and M for mitosis. Growth happens during G1 and G2, but they are actually the gap phases. So if you want to remember it as growth, that's okay, but they were first named the gap phases. And you can see that for, in many cells, a significant fraction of time is spent in growth, but that there has to be separate time committed for either DNA synthesis or for separation of chromosomes. During the G phases, G1 and G2, you're going to have transcription going on, protein translation and so forth. During S phase, you're going to have to stop transcription because you're going through replication. So the cell runs on its stored messages for a little while. And during mitosis, definitely transcription is shut down, and even translation is substantially less than it is during the rest of the cycle. And it's the cyclins that can help control transitions between these different stages. And this is showing you just how some of these rise and fall. Here's one cyclin that's undetectable early in G1, and it rises to a peak near the end of G1. It's overlapping with another cyclin that does extend into the S phase. Cyclin A here is covering the whole of the S phase or replication phase, and other cyclins then cover the other transitions. There are, in a number of cases, cyclins will control protein kinases, and those protein kinases can affect signaling pathways and whether or not they're going to be active or listened to. You're not going to have to memorize these, all right? You don't need to memorize them. But I wanted you to see some authentic examples of what kinds of things cyclins might end up doing. So, for example, here, these are all involved with a phase called start, and start is sort of the cell's decision. I think I'm going to go through replication soon. I'm going to get that stuff together. And so these are cyclins involved in getting ready for S phase. Others that are involved in initiating DNA synthesis. Others that are involved in doing DNA synthesis. And then a little bit of more growth time. The size of G2 varies tremendously with the cell and with the organism. So that can be long or short depending on the organism. This is from a yeast cell. Yeasts grow pretty fast, and you can see a large fraction of their time is in either S or M. Here's some other functions like bud emergence, nuclear migration, cytokinesis, the separating of the cells, and cell separation. So, DNA ligase turns out to be a cyclin in the sense of something that rises and falls with the DNA cycle and so on. During the cell cycle, there are checkpoints in which, in effect, the cell is asking a question to see if it's time to go on and do something else yet. So there's a G2 checkpoint in which questions are being asked like, has all the DNA been replicated? Is the environment in general in good shape? In other words, has it suddenly turned acid? Is it suddenly very salty, whatever? Is the cell big enough? In other words, you need a certain amount of stuff to make a new cell. So these are all functional questions that are addressed by the proteins in the present. And if all the answers are right, it passes that checkpoint, and the cyclin says, okay, go ahead, enter M. Do the M thing. Go do mitosis. And so you're doing mitosis and chugging along. And then there are additional checkpoints. Are all the chromosomes aligned on the spindle? So there's machinery that can detect unaligned chromosomes. And as long as they're not aligned, they're sending up the signal saying, no, wait, don't do it yet. And then when that's been satisfied, that checkpoint is passed, and you can start separating the chromosomes. And so these are all important things that the cell has to keep track of to make sure that during any cell cycle, every bit of DNA is replicated once, and there's enough stuff to make the next round of cell, that the DNA that's been made is intact, and there's no damage going around, that the telomeres have finished elongating, and so on. If these checkpoints send up danger flags, if the answer to these questions is no, it can either stop the cycle at that spot until whatever it is caught up or is taken care of, or it can even trigger apoptosis. It can trigger a programmed cell death. And if the no signal is strong enough, the cell just goes, well, okay, time to get goodbye, and it's gone. Apoptosis is a really dramatic event. It is one form of programmed cell death. It's a way for the organism as a whole or individual cells to say, time out. This isn't going to work. Just bag it. And this is a picture of a cell that's undergoing apoptosis. You can see these little balloon-like things coming off of it. These are little bubbles of cell membrane that are what they say blebbing off. And that they're little bits of the membrane that have escaped from the cytoskeleton. And it's an interesting fact that it's only in little blebs like this that have no cytoskeleton under them that proteins move with a diffusion coefficient you'd expect for the viscosity of membranes. So in a dying cell is the only place where you're going to see proteins behaving like a fluid mosaic. During apoptosis, the contents of the cell get packaged in a way that will attract the attention of macrophages, so macrophages can clear them out of the body. There are some external factors that can trigger apoptosis that are, in effect, death messages from the rest of the body, it's time to go, as well as these internal factors that I've talked about, the DNA checkpoints. Damage to certain organelles can trigger it, and so on. And one part of it is that there are caspases, which are calcium-activated proteases. And since a typical eukaryotic cell keeps calcium under very tight control, if you have a calcium-activated protease, and if things are getting a little mushy and calcium is starting to flow into the cell, you're likely to activate that calcium-activated protease. They have their specific targets. They start attacking those specific targets, and then that becomes kind of a cascade where you get more and more until things are done. Another place in which we see mutations associated with cancer or defects associated with cancer is in signaling pathways. And so the process of coordinating cell growth from the outside, the process of keeping track of differentiation, things like that, there's a little zoo of factors that are involved in cell signaling. And these can involve things like kinases. We saw some of those when we were talking about signal transduction. phosphatases, because whenever you have kinases, you have to have phosphatases, and so defects in phosphatases are comparable to changes in a kinase. Some of these are multi-component systems, so it may not be itself an enzyme, but if it turns on or turns off an enzyme, a kinase or phosphatase. When we look at cell signaling pathways, they don't tend to be straight line chains where this one just goes down to this thing and this one just goes down to that thing. Cell signaling pathways tend to be more like networks rather than straight lines of command. This has the advantage that it makes cells more resilient. Redundancy usually makes for resiliency, but it also makes it a little harder to pinpoint exactly which one specific thing was doing which one thing. But we can say that in a number of cell signaling pathways, these defects in them are found to be present in cells with cancer. So, now one of the important signals is to apoptosis or to escaping to be cancer is DNA damage. That if you have damaged DNA, you'll try to repair it, but if until it's repaired, it potentially can send up a signal that this is damaged, please come do something about it. And so there are checkpoint proteins that can bind to damaged DNA, and in doing so, they will send out a variety of signals. They will recruit DNA repair enzymes. They may up or down regulate cell cycle proteins to arrest the cell cycle. They may trigger apoptosis, depending on which ones we're talking about. One of the important components of these signaling pathways is something called ATM, which is itself a protein kinase. And it can monitor DNA to detect damage. And two of the proteins that it then activates are ones that you may recognize, BRCA1 and P53. P53 may not catch your eye, but BRCA1 is called that because it's the breast cancer gene. BRCA1 is one of the two breast cancer genes that was found associated with higher levels of breast cancer. So ATM, in its normal functioning, would activate BRCA1 and P53. So BRCA1 and 2 mutations in them will increase the odds of breast cancer by 70%. If they are phosphorylated by ATM, this protein that scans for DNA damage, they provide a scaffold for DNA repair and recombination proteins to come in and do their job. If you lose BRCA1, then you're going to have a lot less effective repair, a lot less effective control over the cell cycle. And there's some thought also that BRCA1 may bind to double-strand end breaks to prevent misjoins. And obviously, if you're going to have more of those, you're going to have a higher error rate, higher mutation rate. You need multiple mutations to be a cancer, so that's going to up your chances of getting cancer. Another very common site of changes in gene activity associated with cancer is a protein called retinoblastoma. Retinoblastoma protein gets its name because in infants who are born with two mutations in it are at high risk of getting tumors in the retina. But retinoblastoma protein is present in many places, and its normal function is to hold cells in G1 until the proper checkpoint has been detected. And so RB can bind to the E2F transcription factor. It's one specific transcription factor that's in charge of the cyclins that are needed for the next phase, for S phase. And so as long as RB is active, it will prevent the formation of the cyclins needed for the S phase. We see mutations in retinoblastoma in RB in many different cancers. So this is clearly one of the important checkpoints that's vulnerable, and if it's knocked out, you're more likely to have an escape from proper control. Another protein that we frequently see mutated in cancers is something called P53. P53 has a really boring name. It means a protein that's 53 kilodaltons, so it has a completely trivial name. Sorry about that one. But we know it's important because it's mutated in like half of all tumors. So P53 is clearly one of the key players. And in normal cells, the ways in which the levels of P53 are controlled is by degradation. And if ATM phosphorylates it, the P53 hangs around longer. So this is one of the ways that ATM protects cells that have had cell damage. So if the P53 levels rise, it will delay cell cycle progression. And by holding it back from progressing, that gives a larger window of time for the DNA repair machinery to get to the DNA and repair it. It also has an ability to activate some of the enzymes that are needed for that repair. So it plays an important role in coordinating the cell's ability to repair DNA damage. P53 has sometimes been called the guardian of the genome, which is kind of a cute, grand name for it. And this diagram sort of brings home to you the fact that P53 does a number of different things. And so if you've had some event that's led to DNA damage and ATM is going to activate P53, it can then act on multiple aspects of it. It has a finger in several pies. So it can activate BAX if there's very high levels of damage, and that would trigger apoptosis. At more moderate levels of damage, it might just simply arrest the cell cycle so that there's time for the DNA repair. And it can activate MDM to work on the repair process. So if you lose P53 activity, it's much harder to integrate this response to DNA damage. If you lose P53, you may not trigger apoptosis, for instance. If you lose P53, you won't have as much time to repair damage, and therefore you're going to have a higher mutation rate. So P53 is we can see why half of tumors have lost P53. Losing P53 would put you at risk for a lot of things. This is showing you some of those specifics in the context of response to ionizing radiation, carcinogens, mutagens, a variety of different things. If we look at a cell that's normal and has a normal P53, and these insults, so we remember ionizing radiation could chemically damage things in general, and anyway, we talked about those earlier. So you've got your DNA damage for whatever reason. That ATM steps in, it activates P53, which binds to the DNA. And then the places where P53 is bound to DNA, transcription is turned up for a bunch of different things. P21 is can arrest the cell cycle. GAD45 is a DNA repair enzyme. Those two together can, in many cases, when we're lucky, repair the DNA. In cases where there's high levels or when you can't get the repair, the P53 can activate apoptosis and you die. But let's say we have lost our P53, we still have the DNA damage. You don't halt the DNA, you don't halt the cycle, you don't have time for the repair, you don't die from it, and now what you have are mutant cells. You have cells with mutations of many different kinds. And as that cell goes through rounds of division, the fact that it has some mutations already may contribute to its DNA being unstable and having further mutations. So you end up with a colony that has errors in it and maybe errors that propagate into more errors. Again, you can kind of see where it gets its name, guardian of the genome. Don't panic. This is a picture from my husband. This is trying to convey the integrated network of cell growth and cell death, that you have growth factors and extracellular matrix and hormones and things acting to cause gene expression changes. You have things that will turn down proliferation. And P53 right here is one of the fairly central players in coordinating this orchestra of growth and development and cell death. Another one up in here is RB, and you can see it is also well placed to turn these up and down. So since RB and P53 are both in that orchestra and they're playing different parts, okay, you can talk about sensing nascent tumors or potential tumors. If they trigger it and they say there's problems here, RB activation will shut down the cell cycle and stop growth. P53 activation will separately shut down different parts of the cell cycle, maybe trigger apoptosis or repair. You need to lose both RB and P53 to have a tumor progress. Even if all of that has happened, there's still further things that have to develop for a little colony of tumor cells to become a problem. In the absence of any ability to recruit blood vessels, a tumor is only going to get to be about 1 millimeter. Now, you can see something that's 1 millimeter, but it's not very big. You know, I think if that's 1 centimeter, millimeters like that, you could see it, but it's not very big. It's never going to give you any harm. And it won't get any bigger than that because nutrients can't get in to make the cells grow any further. But if one of the mutations that's happening in these genetically unstable cells is one that turns on the expression of what are called angiogenic factors, then you can start to recruit blood vessels. And once the tumor can recruit blood vessels, A, it can grow, and you can have a tumor that can do harm just simply by its size. And B, it provides an escape route. You see these little cells escaping, so the blood supply that's bringing nutrients can carry away metastasizing cells, and those metastasizing cells can cause secondary tumors that cause further damage. And this is showing that escape in just a little more detail here. Here you have what's called a carcinoma in situ. So it's, those are cancer cells, but it's entirely contained. If in this colony of cells that are all bad in several places, one cell acquires that angiogenic potential, then you get that part is going to get the blood vessels first. That part's going to continue to grow. So you're going to start to get expansion of the tumor. And with expansion of the tumor, you may then get turning on, for instance, the proteases that allow it to escape. And they then get carried through the blood system, and you get metastasis. My husband's a pathologist. He likes his cartoon. It's a caveman saying up here, "It's a mammoth," and down here, early microscope. So, pathologist humor you have to get used to. But I like it. So when we look at cancers overall, we can see that they share characteristics despite their great diversity. They have unstable genomes. That is, they have many mutations, and they go on getting more. The older the tumor, the more different kinds of different mutations it has. They are self-sufficient with regard to growth signals, so they no longer need growth factors. They're insensitive to clues to stop growing, so they're no longer contact inhibited, for example. They can evade apoptosis. They can escape from the signals to die. They have unlimited ability to replicate, so they don't have the senescence and terminal differentiation characteristics that a normal cell would have. They can sustain new blood vessels and blood growth, and they can invade and metastasize. The invasion part means they can break out of their little nidus and dig into another tissue. The metastasis means the cells can go and get anywhere. One particular, to sort of look at these characteristics in the context of one particular cancer, is cervical cancer. This is a cancer of considerable concern. And it's one where we know where the originating cause is, that HPV is human papillomaviruses, cause warts. Most of you have probably had warts on your hands or your feet at some point. A few of these warts, a few of these wart viruses, are consistently present in invasive metastatic cervical cancers that are not just themselves bad, but they can burrow into tissue and spread elsewhere. And we call these high-risk HPVs, high-risk human papillomaviruses. These high-risk HPVs are transmitted almost exclusively sexually, but they are often not known to be there. You may not know that you've had an infection with HPV. We know enough now about cervical cancer to say that HPV infection is necessary for cervical cancer, but it's not sufficient. Other things have to happen in addition to HPV. So that an infection with HPV for over a long period of time can lead to precancerous changes in the cervix. 2% of cancer deaths in the United States are still cervical cancer. In Mexico, it's 25% of cancer deaths. And the large difference here in mortality is due to the fact that there are fairly widespread Pap screenings and cervical cancer screenings in the United States. But this difference here means that in the developing world, cervical cancer is a major killer of women, and at fairly young ages. Many women with cervical cancer leave children behind. So, what is it about HPV that fosters the development of cancer? It isn't sufficient by itself, but it definitely contributes. And so what is it doing? Well, it has a couple of proteins called E6 and E7, which stands for early 6, or early protein 6 and early protein 7. E6 inactivates P53, our friend the guardian of the genome, and E7 inactivates RB, retinoblastoma, the one that could arrest cell cycles. So these two proteins, E6 and E7, take out two of the key agents that are the normal part of the machinery for keeping a normal, healthy cell. And then beyond that, HPV leads to the genome of the cell being unstable, so you tend to get breaks and rearrangements and mutations and stuff like that. This gives you a little bit of a sense of the period of time over which all of these things happen. You remember we talked about the development of diabetes as a matter of time. This is an example of how cancer is a matter of time. And because we know what the initiating causes are here, we have a better handle on the time scale. So from the moment of HPV infection, from quite early on, RB and P53 are lost. Those are going to give rise to the genetic instability. They're going to cause an immortal cell and so forth. But it might be as much as 13 years before the earliest visible signs of this being not a normal cell are. So this is carcinoma in situ and a high-grade abnormality. These cells might be immortal. They are having angiogenesis. They have potential, but they haven't yet started to invade, or they haven't yet started to metastasize. And then after that time, you have the invasion and metastasis. So clearly a matter of development. Okay. See you Friday. Okay. You take over.